All right, today we're going to be going over our PowerPoints. I might look at the manual, and I want to especially show you some things on the YouTube channel that will help you with this week's work. So with this week's work, we're going to go over the nomenclature. That's what we're doing now. At some point during the week, I will assign you a mutant. This will be a mutation, a special mutant allele. Remember, an allele is a version of a gene, just like you have different versions in us that make us taller, shorter, green eyes, blue eyes, red hair, whatever. You have different versions of genes, just like you have two cars. They both may be Camaros, but one has a different color of the car versus another color, but they're still the same car. Well, you have genes that are different versions. That's an allele. And we're going to, you get alleles through mutation. So mutants are are commonly used to do genetic studies in flies, and we're going to assign everybody a mutant, and that'll be your mutant for the semester. So hopefully we'll get to that this week. That'll be coming through both Dr. O'Brien and myself, so this is one where both of us are kind of be working on getting you up to speed on that. We'll also be setting up your first cross. I'll explain that. Now realize WA1 is different from RA1, reading assignment versus writing assignment. I do the writing assignments. You need to listen to me when we'll get the basic instructions for the writing assignment, but how it's going to be graded is very specific to the TA. So realize that WA1 is writing assignment 1, and that primarily comes from me. We may get to the introduction to balancers. Balancers are hard to understand, so I'll go very slow with that. So let's just kind of get on with it on the nomenclature. And we're going to start talking about a lot of terms and things. And so if you're not familiar with a lot of the terms in this PowerPoint, you should go to a specific place on my YouTube channel and get caught up. So I'm going to switch, before I actually even get started here, I'm going to switch to the website here, to the YouTube channel. Now your channel is here. Let's see, if we go to playlists, you have the playlists for Genetics 3311 Spring 21. That's yours. But there's also this one here, this one over here, and you could also just go to all videos, but let's go to the previous semester, and I especially want to point out certain ones. Let's stop that. All right, let's go and talk about specific ones that are going to help out with this set of PowerPoints and understanding the terminology. And if you scroll down, quite a ways down, let's see, there's Genetics Video 3. This one here, I think I made this for an intro class, but it goes through the terminology. What is the difference between a trait and a gene, a character, an allele, a locus, a chromosome? What does the term homologous versus identical mean? All of those, and I kind of draw things along with it. They're not great drawings. I'm not a great artist, but I think this will help you out. So you probably ought to watch this one before you watch the rest of this video. So if you're watching this video, you might want to stop and go watch this one first, because it will help you out. Now another one you might want to watch is up here called chromosomes. Let's see, there's one on chromosomes. Where is that? Did I pass it? I passed it. Crossing over. Chromosome number. All right, right here. CRHOM number. All right, you might want to watch that one as well before you finish these PowerPoints. I would actually definitely recommend both of those. Now, if you're not familiar with the incomplete and complete dominance, you might go here. At some point, you need to learn about codominance. If we don't do it in here, you will need to learn it in regular genetics, the actual course genetics. This is, of course, the lab. There's the regular sheet. You can watch me work through these problems. Okay, in this video, in where I just kind of draw it. Now, of course, I'm not very good, especially doing this with a mouse, but you can watch that. There's also one on a worksheet that I do with my intro class that will help you. Let's see, where is it? There's something worksheet in here, and I've passed it again. Here you might check out. Now these we will do together because they're about FlyLab.js that we do in here. But there are some videos. Now some of these, the audio messed up. I had for a while a bad microphone, so understand that. But there's one in here which says worksheets. You might want to, oh, here it is. Maybe that's this one?
So let's make mom.
Oh, it is this one. But I go through.
Differences will come out.
No, I guess that's.
I guess
that's the one I was thinking of.
And you're comparing the two.
But
in this particular case, you will get different
mapping
answers.
theory,
Right, so
quick
let's do
review. Oh, here, this was last week's quick review of WA1. Maybe you look at that, but realize it might be different this semester. The rules may change. So there are several in here that might help you out for this video before if you're not familiar with all of the terminology dealing with genetics and chromosomes, etc. So understand that. I go on, and the first thing we need to talk about is sex-linked. Realize that it already has two sets of chromosomes. You have 23 pair, 46 total, but one pair are the sex chromosomes, and the sex chromosomes are either X or Y, and if you're female you have two X's, and so they are homologous. You need to understand that term, homologous. But the X and Y are not homologous. So X and X, the two that a female would have, which are two X's, which makes you female. If you have two X's, then you're female. If you have an X and a Y, then you are male, and the X and the Y are not homologous. There are different genes on the X and the Y. On the X, you have the same genes. Now, they may be different alleles. So if you don't understand that distinction between, okay, you have the same genes but different alleles, you need to go understand what alleles are, and that's what those other videos talk about. They talk about the fact that alleles are just different versions of the same gene. And everybody, of course, has two pair of each gene because you have two chromosomes and you have one copy of each gene on each chromosome. You get one chromosome from one parent and the other one from the other parent. You need to understand how that works, and that's what the other videos will tell you. So you need to understand dominance and recessive. There are videos on dominance and recessive. And then what I just talked about, sex-linked. And you need to understand these terminology of the loci or loci. It's just a location on the chromosome where the gene is. So a specific gene will be located on the chromosome at a particular place. That place is called the loci or the loci. Realize that there are strains. Like in flies, we have different strains of fly which have different sets of alleles. Some of those alleles might help you adapt to, say, cold weather. And so you might create a strain designed just for cold weather or just for warm weather. You might design them to not have an immune system. They do that in mice. And you would do that by breeding them and basically getting together a bunch of genes that produce particular traits in the organism that give it certain abilities or lack certain abilities. And those would be artificial strains. And we create those in the laboratory. Especially, it's easy to do with flies. A little harder in mice, but we do it in mice. And so we kind of breed them to have particular combinations of alleles, which are, you can think of alleles as mutant genes. There is the gene that is the most common one, and we generally call that the wild type. So if you see wild type, it's usually a dominant gene, and you need to understand what it means to be dominant. And that gene, if it's the wild type, it's the one that occurs most often in nature. Here we go through what are called, using a Punnett square, and our designation for dominance is a capital letter. The other type of gene is recessive. So you either have a dominant allele or you have a recessive allele. Now, the phenotype, what it looks like, okay, here you can either be purple or white, is determined by the genotype. That's what these letters say, what the combination of alleles you have. So if I have two dominant or capital letters, then I'm going to be whatever that letter is. So big P is purple, little p is white. So two big P's, well, there's nothing but purple there, so of course you're going to be purple. Here I have purple and I have little p here, which is white. So I have purple and white. So I have two different alleles. Well, the capital letter is the dominant one, and it will cover up the smaller ones. So therefore my phenotype, what I look like, is going to be purple. My genotype, I have two different letters. We call this heterozygous. Here, this is called homozygous. We have the same two letters. This is heterozygous. This is also homozygous, homo meaning the same. So I am homozygous recessive because these are little p's. Here I'm homozygous dominant because I have two big p's. And here, because I have one of each, I am heterozygous. But both of these have the same genotype. And that's because the big P covers up the little p. So if you are heterozygous, therefore you end up being purple. You look just like the one with two big P's. The only way to be white is to be homozygous recessive, two little p's. Now down here in our Punnett square, we are looking at what the children will look like if we cross particular parents. So here we crossed a parent, let's see, the male, because here it says sperm, who has two little p's, and the other parent has two big p's. So we cross them and we just bring them down, and this represents the chromosomes. The chromosome have the p's on them. So you get one chromosome from dad, one chromosome from mom, and I strongly suggest to you watching the videos so you understand the relationship between chromosomes, genes, and sperm and egg. All right, these are the sex cells are not the same as the autosomal cells, the regular cells in your body, and they are used to come together. The sperm comes together with the egg, and now the resulting child cells, the child, actually the cells from the organism to be produced, basically has two sets of chromosomes now, and one coming from the sperm and one coming from the egg. And one of those chromosomes has a big P on it and the other one has a little p on it. So therefore this child is big P, little p. And since the father only can give little p's and the mother can only give big P's, all of the children end up being the same when you do when you calculate the Punnett square here. Now in this case, the father still can only give little p. He has little p in his as far as the type of sperm he can produce. But mom in this case is big P, little p, and she can produce two types of eggs. She can produce an egg with a big P in it, and she can produce an egg with a little p in it. So therefore we draw it this way, and because of this change, we now can produce two types of children. Here all the children were purple. In this case, the children, 50% of them will be purple and 50% of them will be white. So keep this in mind. If you are not familiar with this, go back and watch the videos for sure. Get all of the terminology down. So here we go through. You need to understand that you do have 22 autosomes, all right, and then you have one set of sex chromosomes for a total of 23 pair of chromosomes, 46 total. But that last one has the option, if you're male, you'll have an X and a Y. These are not homologous, all right? These are homologous. If you're female, you have two X's. All the little alleles line up at the same space. All the genes line up at the same space, same loci. But the X and Y are not identical, and there are different genes that are unique to males on the Y chromosome. Now, this is fine. You get extra genes. You think, hey, if you're male, you have more genetic information. Not necessarily. This is not necessarily a good case. Realize that you can have bad mutations, and if you have two copies, then basically what you're allowing is you have a backup copy. Let's say you get a mutation here on one of your X chromosomes. Well, you have a backup copy. If this is bad, this is something, a bad trait, well, you have a backup trait. All right, you have a backup copy of the gene that hopefully is dominant over this bad copy, and you don't get whatever the disease is, for instance. But if you're male, you don't have that backup copy, and if you get a mutation on your X chromosome, you don't have a backup copy. You do have some extra genes over here that the genes that make you male, but you don't have a backup copy on these here. So there are a lot of sex-related diseases that only occur in men, because to have the same disease in a female, you would have to have the same mutation on both chromosomes. You don't have that backup copy. And in men, they don't have the backup copy, so they get the disease twice as often as females, actually more than twice the occurrence. So that's the disadvantage of being male, is that you're going to get you're going to be more likely to have these mutations in your X chromosome that will potentially give you a disease. All right. So the X chromosome is much larger. All right, it has approximately 2,000 genes, whereas the Y chromosome only has 78. This has been revised. There are more than that now. We know they were kind of hidden there. But still, the Y chromosome doesn't have a lot of genes. It's very small. Here you can see the size. This is the X and this is the Y, but it does have more than 78. And we're finding that now these genes actually are quite important. For a while, the Y chromosome got a really bad reputation as it was disappearing, and it would disappear eventually altogether. That is not the case. We now know evolutionarily that there are more genes. They're just very subtle. They don't stand out as much, but some of them are very important for the species. The recessive sex-linked trait is to be expressed. Make sure you understand this. For sex-linked, it's exactly what I was just talking about. That females have to have two copies, males only need one. A sex-linked, much more common in males than females. I just discussed that. So understand that, and there are videos out there on the YouTube channel discussing this as well. So as we move through these, we do the crosses. So a sex-linked trait, you don't cross it the same way. I have worksheets. If you actually need more here, I have worksheets to help you with this. So if you want those, I'll go find them and you can work on those. Here, we're going to talk about flies now. And now flies don't have as many chromosomes as we do. They only have really the X, the Y, and then 3 others. So they don't have as many chromosomes. And so you have to understand that their chromosome sets differ from ours. But you still have genes all along here. So all along these chromosomes, this is a way to map it. And these are like the lengths and where they are and which chromosome, 2, 3, and 4, along with X and Y. And so we just have to understand that flies are different than humans. They don't have as much genetic material. In flies, it's still the rate, the ratio of X chromosomes to sets of autosomes that determines the sex in the Drosophila. So the X and Y still determine the sex of the fly, but it's a ratio. It's not like just the presence. And in humans, it's just if you have a Y, you're going to have male features. But in flies, it's the ratio of X's to the others that determine whether or not you're female. It's a little different in flies. And here we have some different types. Now, in my videos, I talk about how this can go wrong in humans, but it's even more complicated in flies. So I would suggest watching my videos on what happens in humans. Then you come over here and realize, oh wow, it gets really complicated because it's all a ratio. It's not like is the Y there. If it's not there, then you're going to look female. If the Y is there, you're going to look male. Your phenotype is male if you have a Y. Not the same way in a fly. And so here, these are the ratios, and this is what you end up looking like, your morphology. So you may look female, all right? Both of these look female, but here you have a meta-female, and I don't have time here to explain, but they have meta-females and intersex. That's another distinction. Let's just put it this way. Fly anatomy doesn't work quite the way human anatomy does, so you can have some really strange combinations here. That's in your textbook if you want to investigate that. We aren't going to work with meta or intersex issues. That's something that's in insects. We're not going to have traits that you have to worry about too much with that. Just know that it's there. You might have it in your regular course, though, the regular genetics course, not the lab. You might need to know at least the terminology. You probably need to know that it's a ratio for this course. Now, in humans, which I give quite a, if you watch my video on chromosomes, you'll get that. Males are what we call hemizygous. So this is when you only have one copy of the X chromosome or one copy of the Y chromosome. So if you have two copies of a chromosome, you are with an allele on it. So you have two alleles of the same type, two dominant alleles. You're homozygous dominant. If you have two copies of the recessive allele, you're homozygous recessive. If you have one of each, a big P and a little p, for instance, you're heterozygous. But with the sex links where you just have one copy, you're not big P, little p, or big P, or little p, you're just P. Maybe you're just a big P. Well, that's hemizygous. Or little p, if you're just little p. And remember, males can be this way. And so males in humans would be hemizygous for any trait that's on the X chromosome. So if you don't understand that, you can ask me questions. I can refer to you other books and videos. You need to understand this. This is Biology 101. You can look this up in any intro book. Tons of videos on this on YouTube if you look it up. I do go over recombination and in the videos online, and we'll be doing more of that later in the semester so that you can understand how recombination works. We go on. Crosses. We're going to be doing what we call reciprocal crosses. We'll be doing double balancer and complementation crosses. We may or may not do the complementation. I believe you're going to have to know how to do them, but I don't think you'll actually have to do them for your writing assignment. So this is just preparing you to get ready to these terms. There's reciprocal crosses, double balancer, and mutant crosses, and then complementation or test crosses. All right. So all of those will be discussed later point. Just get ready for those terms when they come along. Nomenclature. This will get you points counted off on your writing assignments if you don't get this right. So don't forget this when you're writing. All right, the gene symbol, so like big P or little p, as we were talking about, is italicized. So the gene symbol's a shortened version of the full gene name, but either the name, like here, this is dikeet, which its abbreviation is D, but realize they are they are italicized and capitalized, at least in this case. So keep that in mind. You have to get this just right. So curly or CY, this is the abbreviation. This is the full name of the mutation. a lowercase first letter if the mutant is first isolated as a recessive. All right, so remember, little d, if it's recessive, it's going to be a little d. If it's dominant, it's going to be a big D. All right, just more of the same. Just make sure you remember how to do these and how to write it in your paper. If you do it wrong in your paper, you get counted off, and it's actually counted off quite a bit if you don't get the nomenclature right. If you don't write out your genes correctly when you're talking about them in your paper, you talk about the dikete. What was the one before? The dikete. If you talk about dikete and you represent it with a non-italicized D, might be capital, but you didn't italicize it, you're going to get points taken off. And, you know, people get upset, I understand, over something so small. But that's definitely, you'll get counted off for not doing that correctly. All right. There are other symbols like plus. Plus usually designates what I mentioned as the wild type, the type of allele that is most common, and it's usually dominant. So here it's capital C, so it indicates it's dominant, and you have the wild type of this gene. Now 1 indicates that it's not the wild type. It's still dominant, but it's a version, it's the mutation 1. It's dominant, but it's a mutation. It's a different allele, a different version of the gene. All right. Now, this designates you have 2 copies, right? So you could be big P, big P. Well, here you're CY wild type and CY wild type. So you have both dominant genes, and they are both the wild type version. Here you have a wild type and you have a mutant version. So you are heterozygous here. So homozygous, everything is identical on either side of the slash. Heterozygous, you have different versions. All right, heterozygous means hetero means different. So you have a plus here and a 1 here. That means you're different. All right. You'll do some crosses online. I have some extra worksheets. We'll be doing something called the chi-square. That's an issue. It's not easy. I have videos for it, and I will work with you as much as you need to understand it. It's statistics. I teach math as well. I can teach college math. I can teach college computer science. I can teach college biology and bioinformatics. So I teach a lot of things, and one of them is statistics. And it's hard for biologists sometimes to teach statistics. It's not their specialty. I'm good at teaching math. So let me help you. If you get confused with this chi-squared, I can help you. Don't be afraid of it. You can get through doing the chi-squared. I know a lot of people don't like teaching it. I don't mind teaching it. I teach math. Let's see. Just keep in mind that you'll be doing these types of crosses. You'll cross an unknown with a wild type and see what you get. The wild types should be dominant, but you have to do the cross to find out for sure. So you'll get a mutant, and you'll be crossing that mutant fly online virtually. Now, normally in the class, you'd be growing these flies yourself, which is really cool, but we're not doing that this semester because of COVID. You'll have to do it virtually in a computer program. So WT1 is what we're setting up this week, and you'll have an unknown, which you'll just designate it as UN because you don't know what it is yet. The goal of this is for you to figure out what mutant you have, all right, and to figure out if it's dominant or recessive, sex-linked or not, and that can be difficult. But by crossing it with wild types, you can figure out what the nature of your mutation is, and then you can try and figure out, okay, well, the fly has something going on with its wings, so it's a wing mutation, for instance. It's possible, or it could be an eye mutation or something, and they look different. And then you start crossing it with wild type flies, and you look to see what you get. Do you get 50-50 in the children? And then you breed it again, and do you get what do you get? Do you get some other ratio of mutant flies to wild type flies, and that will tell you the nature of your mutation. We'll have to work out how that works, and you'll have to figure it out on your own, and that's what you're going to be writing about in your paper. So you do have to kind of learn the nature of all these crosses. So WT1, and we're going to use it to discriminate between dominance and recessive, or dominant and recessive traits, and you figure out whether or not your wild type was a dominant one or a recessive one, allele. All right, so your unknown unknown or un, and what do you get? Do you get a wild type? Do you get this is for WT2, but we will get there, but just not right away. All right, let's see. How many more? Oh, we've got quite a few more here. All right, so this will determine also whether or not you're sex linked at this point. So we also have to determine if not only is it dominant or recessive, we also have to figure out if it's on the X and Y or Y chromosome. Now here are some new terms that we haven't gone over, and this is a good place to go into this. Expressivity and penetrance. Do not forget to put this in your paper, all right? Somewhere by the time you get to the end, WA5, and we talk about it here, and then you don't talk about it for a long time, you forget to put it in your paper. It needs to go into WA5 somewhere to get the full credit. And I noticed that a lot of people last semester forgot to put in this, so make sure it actually needs to go in all along the way, but it gets counted the most at the end. So make sure that you remember to talk about the expressivity and the penetrance of your mutation. Expressivity is the degree to which the trait is expressed. Penetrance measures the portion of individuals. So this would be like, all right, how red is your hair? So expressivity, maybe you have the gene for red hair, but how red, excuse me, how red is your hair? There are people with really bright red hair, and then there are people with some darker, less bright red. Well, that would be the expressivity. In other words, it shows up to a greater extent in that one individual. Penetrance is a different idea. This has to do with the population, not one individual. This has to do with what proportion of the individuals actually showed the trait. And this is because expressivity can be very low. So if expressivity is very low, they may have the trait, but it actually isn't showing up. So there is not strong penetrance. All right, hold on. All right, hold on, I'm doing a video. Excuse me on that. So be very careful on understanding that the difference between expressivity and penetrance. All right, now writing assignment 1. So this is one that I will grade. Describes the phenotypic characteristics, and it describes the difference between your mutants and your wild types, the difference between males and females. You need to give it a nickname. So you don't know when it's unknown. You don't know what it is. Later on, we'll find the real name for it. But right now, you just have to give it a nickname based on what you found. So maybe the fly has black wings. All right, so you would call it black wings. Or you can come up with a more, maybe it's black speckled. So it looks like you put pepper on your food. So you might call it pepper. All right, so you can come up with a creative nickname if you want for your, and that's exactly where all these names come from, like Dikeet and Curly. Those are all nicknames that became the common name. So we will identify 5 possible genes. You will need to find 5 possible genes that could lead to it. So realize there are more than one gene can lead to the phenotype that you're looking at. Your mutant may, like purple, there may be more than one way to be purple. And so you need to go into the literature and you will find 5 possible, shall we say, mutations that could lead to having a purple flower, for instance, or in this case, curly wings. And you will talk about them in this, this initial part. You will also need to create a figure and along with a photo of your mutant at this point. And you, of course, the photos would come from online. We can't take our own photos because we don't have access to the lab. Otherwise, you'd be growing your own flies and taking your own pictures, but we can't do that this semester. All right, this is the format, so I'm just going to leave this up here for you to read. Make for sure you get it exactly right. If you don't, you get taken off a lot. Okay, the formatting is very important. The content at this point, well, I will count off for you not writing well, but at this point I'm going to be taking off mainly for you getting the format right, because this has to be fixed immediately. You're going to have all your papers will have to be formatted correctly. So I'm going to be looking at formatting on this first one a lot. Your technical content I will count off on, but you have time to fix the technical content. So in other words, if you get something wrong in some of this stuff, I will count off. If you don't, for instance, you forget or don't explain it well, or get it wrong, just plain get it wrong about the life cycles, for instance, you'll get counted off. But the main thing you'll be counted off on WA1 is getting the format right. You can, because this is going to be added to other things to give you WA5. WA5 is the one where I count off for getting everything technically right. Here, you're just getting counted off for the format. Okay? So, in the future, we'll be doing balancers. Now, balancers, much more difficult concept here. So I'm going to go through it quickly, but I have a feeling we'll probably have to do more videos on balancers later. Realize that the chromosomes can get kind of messed up. The genes can move. They can no longer be at the same loci. And that's bad for the fly, but it's good for us doing some of these chromosome tests and figuring out how the chromosomes work and where things are located. And there are known balancers that are known, and we keep stocks of flies, groups of flies that have these genes that have moved. And by crossing them, we can see, it'll tell us how, how certain things work. And so we call these balancers, and there's even a double balancer. So let's talk about how you describe them. F will mean first chromosome, S second, T third, and N means multiple inversions. So what this means is it's kind of like the location. Which chromosome is our balancer on? So the number for different balancers for the same chromosome. So here we're going to, for instance, if a genotype authenticates a slash. So here we have this TM6. All right, that's just on this one. But here we have the slash E1 TM6 would mean that the ebony 1 allele is on a balancer. So this is ebony 1. E1 is ebony. It's a recessive trait because it's a small letter. This is balancer 1. All right, it's just ebony 1. It's the first allele found for the ebony mutation. So it's E1, but TM6 means that it's a balancer. All right, so T, all right, meaning third, M meaning multiple versions, and the 6 could mean several things, but maybe the 6th version of it or something along those lines. So that it means that this allele, this piece, has moved to kind of a new loci. You can think of it this way at this point without me going into more complex description. You definitely need to watch some of my videos on the other from last semester to kind of get an idea of what I'm talking about if this is confusing to you. It just means it's kind of moved. Now, second multiple, 1. All right, the dominant allele is CY1. So what was that? That was curly. And then we have some others. I think this is Cinnabar. I forget what SP and AL1 are. Those are, oh, Aristateless, Cinnabar, and Spec. So these are other mutations. This is curly here. This is and what we're doing here is this is the nomenclature for these particular mutants. Notice that these are all recessive and this one is dominant. See the capital letter. But what we would do is we're kind of showing what has happened here. What has happened is that there's been a rearrangement on the chromosome, and you'll notice that things are inverted. This is not like DNA where it's showing you, you know, this is the other half of the thing. A lot of, for whatever reason, students seem to think that, oh, these are both present and they're related. No, no. What has happened, the chromosome has broken, and it has basically been then reattached in a different orientation. So what happened here is it broke, and so this chromosome is now a balancer kind of chromosome with everything in kind of a reverse order. And this is very useful when this happens. If you need more explanation, just realize this is the normal one, and this is the balancer version, or SM1 balancer. And so everything that was located, for instance, this is 21 to 40, you can see that. Well, now 21 to 40 is like over here going this direction, so it's going in an opposite direction, and that altered how everything works, or how you map it with against another chromosome. All right, this is a wild type fly, and here is your balancer. It's got a double balancer actually. It's got a CY and a PM, both of them dominant traits, and then it has another set of mutations. It has D and SB. I don't know what these are. I know this one's curly. I think this is plum, dikeet, and oh, I've forgotten what SB stands for. Cinnabar? No, that's CN. But it has 4 mutations. This is the wild type. And you can see you add these mutations, this fly looks a lot different, right? The wings especially, the abdomen's a little different. There's some maybe it's a little shinier here or something. This is a mutant fly. This is the wild type fly. All right, that just to show you kind of the idea of what happens when you get mutant flies. Here's wild type, and here's one with stubble. So you can see this one has more hairs on its back. Or whatever, more long hairs anyhow. This one just has them down here, not so many up here. And so this is stubble. This is a mutation, and they call them thoracic bristles on the scutum or scutellum, and that's just the location here, the back. So on its back, this one has a hairier back than this one. That's kind of the way you can say that in non-technical terms. All right, we're getting close to the end, I believe, here. So here's wild type, and oh, here's another version of stubble. It also has longer hairs on its head. So the head bristles are different in the mutant. And here we have, what is this? Plum. I know that's plum here. I think that has to do with eyes. Maybe not. But you can see that is where the mutation is here. It's in the eye. This is a wild type. Should have this nice spot in the middle. We'll see how it's faded across the whole thing. And this fly, I believe, doesn't see as well as this one. Doesn't focus as well. All right. Are we done here? I think that's it. Those were our examples. Watch some of the videos and be on the lookout for more videos all week. I try to send out an email when I put a new video up, but not always. So check every now and then for videos, and watch the ones that are up from last semester. Feel free to watch any of the videos on there if you feel you need a review. Oh, one more thing before we go. I did hand out the the laboratory manual, so make sure you take a look at it. It has a lot of this material in it. Uh, all the things that you need to be doing, the nature of crosses. Let's see, how you score things. This is a great how to interpret what these little tables are saying. Double balancers, how you should word it. So make sure you review this manual. That also is coming up, this week. So everything is here. This is the basic procedure. Of course, we can't do these in the lab. We have to do them with software now. And make sure you do review all of this. Here, this is very important. Figuring out how this table works. I'll probably have to go through this and make a video on how to read this table, but try to read this table and figure out how this process works. Let's see, same here. Further down, here's where we get to the chi-square. If you have any problems with chi-square, talk to me. I can explain it. I have videos already out there on chi-square, but I can do it with you. Here's the template for doing the chromosome diagrams. You can use this or make your own. You'll figure that out at some later point. But make sure you go through this, the laboratory manual here for Project 1. Email me if you have an issue. Show up on Fridays for the meeting. At this point it's at 4:30. I'll send out an email right beforehand giving you a link. But kind of be watching for the email maybe, or if you already have the link, it'll be the same as before, but I'll be online at 4:30 unless something changes. Sometimes I have meetings I have to go to on Zoom myself, and they tend to put them on Fridays, and if one of them comes up at 4:30, I try to put it at the end of the day, so all the other meetings are done, but they may not be done, so I may be in a meeting at 4:30. If I am, I will reschedule.